Hey everybody, welcome to week 8 of Physiological Psychology from Dr. Kirby's Living Room. Here we are at midterm, and we are getting deeper into some more specific aspects of physiological psychology. This week, sensation and perception. For this week, I would like y'all to read modules 5.1, 6.1, and 6.2 in your textbook. I will also be covering a little bit of the visual information from 5.2 and 5.3. I'm trying to make sure that for 5.2 and 5.3, you get all the information you need here in this lecture and some of the supplemental videos without having to have an overwhelming amount to read this week. And then read 6.1 and 6.2 and complete Prep Guide 6, our final one before our science communication project. Let's start with Chapter 5, all about vision. We are going to talk about visual coding, which is from 5.1, processing visual information, so that's a little bit from 5.2, and then parallel processing with a little bit more from 5.3. So to begin visual coding, I'd like you to watch one of the supplemental videos here. That video about the eye and the brain hinted about some of how we see color vision, but now I want to dive deeper into the 3 theories your textbook uses to explain color vision. You think it might just be a matter of examining what the eye is made up of, what the brain responds like, and then we would know exactly how color vision works. But our understanding of it has evolved over time, and before we had the technology to know exactly what all these cells and brain areas were doing, we had to have some theories about it. The first is the trichromatic or the Young-Helmholtz theory, which was first posited back in the 1800s before we had any way of really measuring what was in the human eye or the human brain. This theory states that the reason you see color is because you have 3 types of color receptors in your eye that respond to different wavelengths of light. So basically they pick up different colors, so to speak, of light. And when you see a color that's a mixture, it's because some of your bluish receptors and some of your reddish receptors are activated at the same time from these wavelengths of light, and that's going to show up looking like purple to you. So throw everything, for all these 3 theories, by the way, everything you learned about what primary colors are and how to mix them and stuff like that, totally out the window, because the science of the eye does not work that way at all. So what we're looking at here on this graph are the wavelengths of visible light in the spectrum. We have the shorter wavelengths are more blue-like and the longer ones are more red-like. As we go towards the right on that x-axis. On the y-axis, we are seeing just how quickly those cones are going to respond, meaning like how many action potentials they're going to send. So as that curve goes upward, that means that at that wavelength of light, the cones are firing the fastest. They're responding the most strongly to that wavelength of light. So we do have 3 types of cones. Helmholtz figured this out after Young had just theorized it years before. We do have 3 different types of cones in our eyes. There is the short wavelength cone, which is, that's what we mean by S here. And there's the medium and the long wavelength cones. This does not mean the size of the cone itself, short, medium, and long. It means the size of the wavelengths of electromagnetic radiation that they respond best to. So we don't have blue, yellow, and red like the primary colors. We actually have the short wavelength cone responds most to bluish colors. The medium wavelength one responds sort of to yellow and greenish stuff, and then the long wavelength one overlaps with the medium one a lot, but it's more in the redder phase. There is actually a kind of rare mutation that tends to be a sex-linked trait that shows up more in women, where they have a 4th cone that's even further into that red field, so they can distinguish between reds and pinks that the rest of us with only 3 cones cannot see the difference between. But most of us only have these 3 cones. And so, if you see something that's green, it's because those wavelengths of light are close to the 550 nanometers in wavelength that bounces off the object, hits your retina, and some of your cones are not going to do anything when they receive that light. They're not going to really fire at all, like the short wavelength ones. And some of your long wavelength ones might respond somewhat. But let's say it's more like 500, closer to the bluer. You're going to have, let's say it's like around 525, pretty firmly in the green and not overlapping with that red one very much. You're going to have most of the medium wavelength cones firing pretty rapidly. Now, if you see stuff that's in the yellow range, which is between the green and the red there, you're going to have some of the green ones and some of the medium and some of the long wavelength tones activated at the same time, and that's going to contribute to your brain perceiving the yellowish color. So Young-Helmholtz theory is based on you have three cones, and we're basically mixing together three types of colors in order to see the range of colors that those of us with cool color vision do see. But there's some weird phenomena that Young-Helmholtz theory cannot explain, so we're going to demonstrate them. Let's try this visual illusion. You might be able to see that there's a tiny white dot in the very, very center of this weird colored flag. I want you to focus on that dot, don't move your eyes, and focus until I tell you to stop. So we're going to go for around 30 seconds. You may already notice some things about this picture changing. Okay, 30 seconds is up. What do you see now when you look at the blank white one? So, you probably saw something that looks like this. We used the inverted colors of the American flag. We focused on them for a long time, and then when we looked at a blank white screen again, we saw the red, white, and blue, and the stars and stripes. How and why does this happen? The opponent process theory was developed to explain these negative after images, which is what you might have just seen if this demonstration worked for you. So this bipolar cell on the left receives input from the activity of those short or blue cones. And when you see blue and those cones are activated, they're sending an excitatory message, and this bipolar cell sends an excitatory message for blue to the brain. When you're not looking at blue, it's sending an inhibitory blue message, which is kind of actually like sending yellow, which is blue's opposite. When you receive excitatory message from your red or long wavelength cones, that's going to contribute to your perception of red. But when you're not looking at red, when you're looking at green, your bipolar cell is sending an inhibitory message to the brain. So if it's inhibiting red, it's going to show up as blue for you. It's paired opposite. Now, the fact that red and green are paired together explains why a lot of colorblind people mix up these colors. They can't tell red and green apart because they're missing the red cones. Everything's going to look kind of greenish, bluish, yellowish to them, and they're not going to even be able to see red because these bipolar cells can't send an excitatory message from those damaged or missing red cones. And then for the white-black one, this bipolar cell is going to send an excitatory message to the brain when you get input from all your cones mixed together pretty much equally. So we know that black is kind of like all colors mixed together, right? And then when it's inhibited, you're going to see white. So imagine that when you saw the yellow square with the black stars in it on that flag, and then you looked at the blank white, your blue-yellow linked bipolar cell was sending an inhibitory blue message and thus an excitatory yellow message to your brain, so you're perceiving yellow. So that contributed to you seeing yellow and inhibiting blue and yellow colors. And then when you look away, your receptors were now fatigued, and so the inhibitory signals take over. Blue was being inhibited, but now your receptor is kind of tired of inhibiting blue and sending yellow, so it does the opposite. It inhibits yellow and sends blue, and that's why you saw the blue square with white stars. So basically, what you need to know about opponent process theory is that some colors are kind of paired together due to excitatory and inhibitory signals from cones being sent to the brain. So it's not just cones, it's the relay between the cones and the brain that help you see these negative after images when your receptors get fatigued. But that's not the only aspect of color vision that we need to explain. This theory, sort of like Young-Helmholtz theory, didn't quite go far enough. So let's look at another demonstration to show what gap the opponent process theory still has that it doesn't explain, and how we have a new theory to explain other aspects of color vision.